Hello guys, it's my hope that you're doing well. Welcome back to Miss Fountain channel. In this session, we're going to look at factors that can complicate the interpretation of DNA profiles. In one of our previous videos, we looked at DNA profiles and how they are generated, the different ways of generating the profile. And this is like a continuation of that video. So we're going to look at the factors that can make the interpretation of those DNA profiles complicated. And in this we have different factors. We have about 5, which are stutter, spike peak, we have degradation, we have blob or background noise, and we also have pull up. We're going to start with stutter. Stutter is sometimes referred to as little extra peaks. And stutter peaks are small peaks that occur immediately before or after a real peak. And during the PCR amplification process, the polymerase can lose its place when copying a strand of DNA, and this is usually slipping forwards or backwards for base pairs. And the result is a small number of DNA fragment copies. So we refer to as stutter peaks. So these will be extra peaks that are either immediately before or immediately after the real peak. The next factor that can complicate is degradation. DNA samples degrade when a sample is aged or even exposed to harsh conditions. And degradation is usually marked by progressively falling peak heights as the amplicon size increases. Degraded samples are sometimes difficult to interpret because of the loss of peak height information. Another factor that is difficult to explain is peak height imbalance. And peak height imbalances are calculated in heterozygous loci, that is one that contains 2 alleles. A peak height imbalance is generally considered to occur when there is a greater than 30% difference in the heights of the 2 peaks. That is, the 2 peaks differ with more than 30%. A sample from a single person should contain peaks that are roughly equivalent in height. That is, the difference shouldn't be that much. So an imbalance is an indicative of a mixture. Another factor is blobs and noise. Okay, we found that during the testing process, excess dye can clump together and result in a dye blob appearing on the electropherogram output. Although dye blobs can be observed at many heights, most tend to exhibit a wide shape. And the danger of dye blobs is that they can mask a true allele. Noise is usually present in all samples and constitutes the small peaks along the baseline. Sometimes noise can be a problem if it's too large. Air bubbles, urea crystals, as well as sample contamination are among the possible causes of large peaks that may mask contributions made by other contributors or may be mistaken as contributions made by a victim or even a suspect. Another factor that can challenge the interpretation of DNA profiles is pull-up, and pull-up is sometimes referred to as bleed-through. And this is a failure of the analysis software to discriminate between the different dye colors that are used during the generation of test results. A peak observed is in one dye, such as blue, is recorded by sensor for another dye, such as green or even yellow, and generate a second peak that is a technical artifact. We also have another factor being spikes. And spikes are technical artifact peaks that are caused by voltage spikes during analysis. So when there's a voltage spike, there'll be technical artifact peaks, and these peaks are referred to as spikes. And spikes typically exhibit an unusually thin peak shape. As well as technical artifacts, the danger is that the peak will go unnoticed and will potentially impact the DNA testing results. So those are some of the factors that challenge or make the interpretation of DNA profiles a little bit challenging. We've said that we have spikes, we have pull-up, we also have blobs in noise, we have peak height imbalance, degradation, as well as stutter. Thank you for watching this video. Remember to like, comment,